Okay, hello. Um, oh, okay. All right. Okay. So last lecture, I finished talking about what enzymes did and how they worked. Okay, this one, we are going to talk about everything else dealing with enzymes and how they catalyze reactions and stuff like that. Okay. Okay, so there are enzymes. Enzymes do almost everything, but we tend to put them in categories of what they do. Okay, so okay, phosphorylation is the addition of a phosphate group. Okay, oxidation reduction reactions. Um, you probably learned this in chemistry or whatever form of chemistry you took. Um, I think we have a scientific concepts class, and you guys probably learned this in scientific concepts. Okay, so um, oxidation reduction, these two always happen side by side. So one thing gets reduced, one thing gets oxidized. Okay, the one thing that is oxidized gains, right, the one thing that is oxidized loses electrons. The one thing that gains electrons is reduced. Okay, so the easy way to remember is oil rig. Okay, oxidation is loss, reduction is gain of electrons. Okay, the same can be said for energy because electrons contain energy. So anything that's reduced gains energy. Okay, anything that's oxidized loses energy. Okay, that's going to be a very important concept in just a couple of slides. So make sure you understand that. Okay, electrons contain energy. So if I gain electrons, I gain energy. If I have to give away the electrons, I'm giving away packets of energy. Okay. Um, okay. Hydrolysis and dehydration, these are in the opposites of each other. Um, dehydration reactions, the water is a product. Usually the concept is dehydration synthesis. Okay. I'm taking this thing A, and I'm attaching it to B, and I come out with A being attached to B, and I have a water. Okay, that is a dehydration reaction. I am getting water out as a product. I'm dehydrating a water. I'm removing the water from A and B here and using it to put it together, and I come out with water. Okay, if I run this reaction in reverse, so I'll show this in red. If I run this reaction in reverse, okay, I am hydrolyzing. Okay, hydrolysis. I'm taking water, I'm shoving it in between A and B. So now I have A and B, and I lose the water. Okay. Okay. Um, addition subtraction reactions. This is just you can read through it. You should know what each one needs to do. Addition reactions, subtraction reactions. There are things that can fit in multiple categories, like kinases. Okay, a kinase is something that adds a phosphate group, so it's an addition reaction as well, right? Animation as a okay, aminases. Okay, add a amino group. Okay, so it's also an addition reaction as well. Okay, synthetases are like ligation reactions are where they join two things together, like two bigger things, not just a amino group or a phosphate group, but an entire peptide together or entire stretches of DNA together. So synthetases. Okay. All right. So make sure you understand everything. We used to all this time explaining table 4.4. So make sure you understand table 4.4 and what to do. Okay. All right. So metabolism. Okay. Metabolism is technically all the chemical reactions that occur within a cell. In you, metabolism is your bunch of cells, so metabolism is all the chemical reactions that take place inside the organism. Okay? Okay. Metabolism breaks up into two components, catabolism and anabolism. Okay? Catabolism is when you break apart things. Okay? You catabolize things. Okay? You take things, you break them apart into smaller bits. Whenever you do that, whenever you break things into smaller bits, you usually release energy. Okay? That energy is used to fuel the second half of metabolism, anabolism. Anabolism is taking the energy that you stored and using it to build things. Okay? Like muscle or new cells, okay, or fat. Like all of that is anabolism. Okay. Okay. Metabolic pathways resemble a roadmap. Okay. So this is what metabolic pathways actually really kind of look like. Okay. Actually, I'm going to have you take a look right here. This is what metabolic pathways actually look like. Okay. The way we present it in class always is a very simplified version. You guys are not training biochemists or anything, right? So this is what it actually looks like and what is actually happening. All right, so back to screen one. Okay, so here, okay, this is what metabolic pathways kind of look like. The way we teach you guys is actually a even more linear version of this, but do understand there are byproducts and different branching points that we can go off of and stuff like that to do other things. Okay. All right. Cells regulate their metabolic pathways. It shouldn't come as a surprise to you, but we can't just have everything occur all at once, all at the same time, with no particular reason, rhyme or reason, right? So we need to control our metabolic pathways. Okay. We turn some on, we turn some off, and depending on our cellular needs at that time. Okay. We do that primarily by controlling enzyme concentrations. So if we want more of a chemical reaction, we make more of that enzyme. Okay. We can also put modulators on the enzymes themselves that make them work faster or work slower, mainly work slower. Okay. we have different enzymes to catalyze different reversible reactions. So if we want a pathway to change direction and go backwards, we can make the enzymes do that. Okay. Compartmentalizing enzymes within organelles. This is a big deal. This is why we are more complex than bacteria. Bacteria cannot do this. We can because we have different compartments inside our cells, and therefore we can have different compartments with different enzymes in it. So if we want something to work faster, we send it to that compartment. Okay. 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 A very, very important concept that you guys learned in chapter 1 was negative feedback. Okay. Negative feedback works in cellular pathways as well. Okay. So here is a pathway. Okay. So A goes all the way through to Z, Z being the final product. Okay, if you have enough of this final product, you do not want to make more. Okay, you always want just right amounts of everything. Okay, so if you have enough of Z, you don't want to make more of it. So if you don't want to make more of Z, then you don't really want B and C either, because those are just intermediates that will lead to Z. So what you want to do is inhibit this whole pathway from working at all. Okay, and the best way to do that is to have Z feedback on A. Okay, so if you have enough of Z, it will block enzyme 1 from working. Okay, these parallel bars here, or this perpendicular arrow, okay, means that this is inhibiting enzyme 1, is preventing enzyme 1 from working. If you have no enzyme 1, okay, then you have A, but you can't turn A into B. So you stop this entire pathway from working. Okay, this is all great until you run out of Z. If you run out of Z, then you don't have Z inhibiting enzyme 1 anymore. So A is now able to turn into B, B is able to turn into C, and eventually you get more of Z. And then you have enough of Z, and then you inhibit A, and now this pathway no longer works. So this pathway only works when you don't have enough of Z. So you always want to make just enough of Z to get by. Okay. Okay. So most enzymes actually catalyze reversible reactions. Reversible reactions are so you can do this back and forth very easily using the same enzyme, carbonic anhydrase, and you can switch CO2 to water or CO2 plus water into carbonic acid very easily. Okay, because these are reversible. Their delta G's are very close to 0, so they're reversible. Okay, you can have reversible reactions like this, where you have 2 enzymes, one catalyzing the forward reaction, one catalyzing the backward reaction. Okay, and these are irreversible reactions. Okay. I know it's confusing that using the same enzyme for the series, but in reality, this reaction they're showing you is actually an irreversible reaction. You have this enzyme that go back, but it actually, this enzyme requires ATP. This one that goes back does not. So it's technically an irreversible reaction. Okay, they're showing you 2 different enzymes can work on, like one can make it go forward and one can make it go back. Okay. All right. I'm sure you guys know what ATP is by now. It's one of the few things that I'm almost certain everybody learns in high school biology. Okay. If you haven't learned it in high school biology, definitely learn it in anatomy and microbiology at MSWU. Okay. Okay. So ATP, okay, is the high energy molecule. Has a high energy phosphate bond. Okay. Um, okay. So one glucose molecule can yield about 30 to 32 ATP. Okay, so every molecule of glucose you eat can yield about 30 to 32 ATP. Okay, in aerobic metabolism. In anaerobic metabolism, when you have no oxygen, it can only make 2 ATP. Okay, there are catabolic pathways that produce ATP, and so and their glycolysis, the citric acid cycle, and the electron transport system or the electron transport chain. Okay, so we use these three pathways together to maximize ATP use for aerobic metabolism. Okay, when we calculate out the ATP from glycolysis, from the citric acid cycle, and from the electron transport chain, the number turns out to be 38. Okay, 38 ATP is the ideal number. Okay, like if everything works perfectly, you get 38 ATP. But it doesn't. Like your cells do not work perfectly. Okay, so on average, the yield is actually about 30 to 32 ATP, which is why they present that here. Okay. All right, so here is how we make ATP from glucose. Okay, so ATP goes through glycolysis, goes through the citric acid cycle, and then goes through the electron transport chain. Okay, so let's go through each step one at a time. Okay, so here is glycolysis. Okay, glycolysis. Okay, here is what's going on. You're taking glucose, so this is glucose right here. You are initially using up a couple ATPs. Okay, so you have to prime the pump a little bit. You have to add some energy in, but when you finish through all this, you get more energy out of this. Okay. Pause this for a second. All right, so I enlarge this to make sure you understand. So make sure what I'm gonna make sure you guys understand are the key features. Okay, and like don't memorize every single step. Okay, that's not relevant. What I want to make sure you guys understand is like all the stuff that's highlighted here, and the inputs and outputs. Okay, what goes into each step and what comes out of each step. Okay, so oops. Okay, what goes into each step. Okay, and what comes out of each step. Okay, all right. So and make sure you follow the NADHs as well. See these little explosion things? That means they have high energy. We're going to cash in all these high energy bonds later. Okay, so make sure you go through this again with the citric acid cycle and the electron transport chain. You'll see like all these little things. I want to make sure you guys understand like the ins and outs of every step of the process. Do not spend your time memorizing enzymes. Okay. 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 So here is acid cycle. Okay. You can read all about acid cycle here. Okay. And this is why we have mitochondria in the first place. This is mitochondria, and this is where it actually takes place. Okay. So if you go through the citric acid cycle, make sure you're aware of how many NADHs are released here and here. Okay, how many FADHs, how many ATPs. Okay. Okay. And then last but not least, how the electron transport system actually works and how we get more ATPs here. Okay. What is the need for oxygen here? This is why we need oxygen. This is why we need to breathe oxygen so we can fuel this reaction. Okay. All right. So you can read the book on how this works. Okay. All right. So, okay. So let me talk you through this on a white screen. Okay. So let's start. Okay. Sugar is C6H12O6. Okay. Uh, this is glycolysis. Okay. Okay. So to go through glycolysis, I'm gonna take sugar and I will break it down into pyruvate. Okay. I will split that sugar in half, glycolysis, right? Glycolysis, I'm taking a glucose, glyco, and lysing it open, splitting it into two. Okay, so I get two pyruvate. In addition, I get two ATP. Okay, I remember I used two ATP in the beginning. I primed it with two ATP. In the end I get four ATP back. So total overall I get two ATP increase. Okay. All right. So that's two NADH. Okay. These NADHs, okay, are high energy molecules. Okay. Anytime I add a hydrogen, there are electrons that come along with the hydrogen. Okay. So if I see more hydrogen on a molecule, that means it's more reduced. Okay. That means it has more energy. We're going to cash in the energy later. Okay. All right. So glycolysis, I am taking sugar, I'm breaking down into pyruvate. Okay. And then I'm breaking this down into two NADHs. Okay. So now I have more energy. Okay. So, oops. What the? Okay. Sorry. Oh, okay. Okay. Sorry about that. I don't know what the hell's glitching here. Um, let's change color. I am going to take pyruvate, okay, and I'm going to use it. Okay, and this is now the citric acid. Okay, I'm going to put this in blue so you can see the individual steps. Pyruvate goes to 2-acetyl-CoA. H plus 2CO2. Okay, so the citric acid cycle has two little steps. Okay, the first one, I'm going to take the pyruvate and break it down further. So I'm going to take pyruvate and break it down to acetyl-CoA. Okay, get 2 NADHs out of it and 2 CO2s. Okay, these CO2s is going to be what I breathe out when I breathe out. Okay, the 2 NADHs. So I'm going to use the same thing here. So now I have 4 NADHs. Okay, these NADHs are going to be very energetic. Okay, and we'll cache them in later. Okay, the next step. Okay, I'm going to take acetyl-CoA. Okay, we'll take 2 acetyl-CoA. Acetyl literally means it has 2 carbons. So acetyl-CoA. And we'll break that down. Okay, plus 4 CO2s plus 6 NADHs plus 2 FADH2s. Okay, in the diagram in the textbook, okay, it shows you like what happens for one acetyl-CoA. Remember, in glycolysis, we broke one sugar molecule into 2. So I'm keeping the 2 here. So everything's multiplied by 2. Because you want to, eventually you want to sum it up and figure out how much ATP is made by one glucose molecule. Okay, so here is the acetyl-CoA. Acetyl-CoA breaks down to CoA, which we recycle later, and then 4 CO2s, 6 NADHs, and 2 FADHs. Okay. Okay. So now, turn this into black. Okay. So total so far, we have, oops, I forgot a little something here. Plus 2 ATP. Okay. So we have 4 ATP now. 4 ATP now plus 2, 2, 6. Okay. So that is 10 NADH plus 2 FADH2. Oops. Okay, so that's the total we have so far. This, the final step, is going to be black. Black. Okay. Electron transport system. If you hear me talk about an electron transport chain, they mean the same thing. I'm just used to using the word chain. Okay. Okay. So the ATP we can already use immediately. The NADH and FADH2s we cannot. Okay. The way the electron transport chain works. Okay. What we'll do is the 10 NADHs will eventually make 3 ATPs, or 3 ATPs each. Okay, so each there's 10 of them, so 30 ATP. The 2 FADHs will eventually make 40. Okay, so if you add 4 to 30 to 4 here, you get 38. Um, 2. Okay, hopefully you guys understand all this. Okay, if not, ask me in class and we'll go over this. All right. I'm gonna end this recording here, and then there will be a part 3 that goes over how the DNA code works. Okay. All right. Okay, everybody. Sharing.